The effects of adding saponin into microbial fuel cells. Hi, my name is Skylar Choi, and I'm here to alert you of a current possibility. I'm sure most of you will agree with me when I say that climate change is becoming an increasingly imminent issue within our society that will gradually take more control of our lives, from simple weather changes to possibly more frequent natural disasters. For those of you who don't know, climate change is the gradual changing of climate due to the increased levels of atmospheric greenhouse gases such as methane and carbon dioxide produced by the use of fossil fuels. And according to NASA, for millennia, atmospheric carbon dioxide levels had never been above 300 parts per million, but our current level is above 400 parts per million, and the level is only increasing at a rate so high we've never seen before. But don't worry, solutions to combat the climate crisis include the use of alternative energy, which is basically energy produced elsewhere than fossil fuels like energy from wind turbines and solar panels. Microbial fuel cells, MFCs, are being heavily researched as a new form of alternative energy. Essentially, the way these work is that bacteria like Geobacter and Schumannensis catalyze electrochemical oxidations or reductions at an anode or cathode respectively to produce an electric current. But can MFCs be put into use? Can they generate enough power to be considered usable? Is it possible to scale up the use of MFCs to industrially relevant capacities? The answer to these questions are currently no. So in order to be able to take advantage of this amazing technology and not put it to waste, there must be some change. Now you may be thinking, well, why use MFCs at all if there are other sources of alternative energies? Why not use those? The advantage of using MFCs is that they can be used towards wastewater treatment, unlike the other methods, which cannot. They are also less dependent on renewable energy technology, and they can be built fuels. Today, I will describe a possible way to increase productivity in MFCs, but before I do that, here's a timeline of what I will go over. Methane is usually produced in anaerobic environments such as soil, sediments, and waste treatment plants. The usual pathway for methane production is a carbon dioxide reduction involving hydrogen as a reducing agent. The hydrogen fusion rate is slow, and this decreases the rate of methane generation. Methanogenesis, which involves the consumption of electrons, happens at the very end of the methane production process and accelerates it. Methanogenesis relies on a process called direct interspecies electron transfer, otherwise known as diet, which does not rely on the diffusion of gaseous compounds and instead fosters the syntrophy between electron donating and electron accepting partners. It has been concluded that all microorganisms are capable of extracellular electron transfer with the electrodes with the ability of diet. Since the electron acceptor is a microorganism instead of an electrode in an MFC, it's really reasonable to occur an electrosyntrophy process between bacteria commonly used in MFCs, such as Geobacter sulfurducens and Schumannensis that produce methane. Methanogenesis happens in MFCs and consumes electrons because it diverts electrons from the anode. Thus, methanogenesis serves as competition for power generation for electrons. Organic matter such as Korean ginseng and bellflower roots have shown the inhibition of methane production and a consequent increase in electricity generation in MFCs. The active components in both ginseng and bellflower roots are thought to be saponin. However, saponin has not shown any direct methanogenesis inhibition, but has been associated with the indirect methane decrease in open systems with less methane production per animal body weight. The MFC anode chamber is a closed system for anaerobic fermentation, and methanogenesis occurs there. Thus, methane inhibition is definitely expected there with saponin supplementation based on the fact that it worked with toraji. These are the primary articles I use, and a full list of references is detailed at the end. This is an overview of microbial fuel cells, which talks about the advantages. For example, the MFCs can be used for wastewater treatment. It also talks about the limitations of MFCs, low power for example. And lastly, it focuses on enhancing the performance of MFCs by acknowledging the problem of slow rate of electron transfer to and from the anode of the MFC, which affects power output. This article describes the direct interspecies electron transfer, otherwise known as diet, that methanogenesis relies upon. It essentially fosters the syntrophy between electron donating and electron accepting partners in MFCs. The main purpose of this article was to show that methane production consumes electrons because of an electrocentrifugatory practice between bacteria and MFCs. This article associates saponin with indirect methane decrease in organic matters, with jamun being the organic matter containing saponin in this case. This article also associates saponin with the indirect inhibition of methanogenesis in organic matters and links it to an indirect increase in electricity generation in MFCs, this time with Korean ginseng and Korean bellflower dodaji roots as the organic matter containing saponin. Also, it shows that methanogenesis occurs in the MFC anode. And for this last article, we will get back to later. To recap what we know so far, number 1, a flow of electrons makes up a current. Number 2, methanogenesis consumes electrons. Number 3, methanogenesis may compete with power generation for electrons. Number 4, organic matters containing saponin showed indirect methanogenesis inhibition in various systems, including MFCs. However, there remains a gap. It has never been proven directly. Yes, saponin has been associated with indirect methane decrease, and if methanogenesis were to happen anywhere in an MFC, it would be within the anode, but nobody's tested this, so even though theoretically it is known that saponin would correlate with increased electric generation in MFCs, nobody knows the extent of the increase. Nobody knows whether there will be more that must be done besides the addition of saponin for MFCs to generate enough power to be considered usable, and nobody knows whether the amount of saponin added into an MFC correlates with better productivity. Which leads us to the research question. Of course, not everything can be tested at once, and I am deciding to test the extent of the increase in productivity and whether it will be significant enough for MFCs to generate enough power to be considered usable. In my experiment, there will be 2 groups of MFCs. Group A is control and will not receive any saponin treatment, and Group B will receive 4 milligrams of saponin. Each group will consist of 3 MFCs. To validate the consistent effects of treatment, statistical analysis will be performed and experimental significance will be declared at P is less than 0.05. All MFCs will be filled with 100 milliliters of rumen fluid inocular and 2 grams of cellobiose, and they will all be incubated at 37 degrees Celsius in a water bath. But why are we using rumen? For this article we skipped over before, it describes why we are using rumen. We need 2 kinds of bacteria, one that is electrochemically active, which transfers electrons to the anode, and the other that is cellulotic bacteria that degrades cellulose. Rumen fluid is thought to have both. The amount of methane produced will be determined by estimating organic matter degradation, and that is found by measuring the volume of gas production, the volume of gas composition, and volatile fatty acid production. The electric current over the 430 ohm resistor will also be measured after 3 days of buildup in bacteria in MFC. The electric current will be measured twice a day until day 5, and if electric current values are the same or similar between MFCs on day 5, treatments will be added to the MFC. Disclaimer: organic matter degradation and electric power generation by rumen fluid inocular itself in all groups is uncontrollable and may change and differ for each rumen fluid inocular collection. The use of the same inocular allows this uncontrollable factor to be considered as a constant variable, and the difference between control and treatments will prove the effect of treatments. And the expected result is that there will be a direct correlation between saponin addition and methanogenesis inhibition, but what we're looking for here is whether the saponin added will increase the productivity level of the MFCs to a level where they will be able to be used in the real world in wastewater plants. Currently, MFCs have an extremely low average power output range, ranging less than 0.1 to 1 megawatts, while other alternative energies have an output range from 0.1 to 10 megawatts in the case of wind energy, and in the case of hydroelectric energy, the range goes from 0.1 to over 1,000 megawatts. Now, saponin will definitely increase the efficiency in MFCs and thus the power generation. And the goal is for MFCs to expand the range of the average power output to 10 to equate the higher end of the range power output of MFCs to other sources of alternative energy, such as wind energy and energy from phosphoric acid fuel cells. Then MFCs will be deemed productive enough to help with wastewater treatment. First, to recap. MFCs are, of course, of paramount importance to society in the midst of this climate crisis. Let's go over the advantages. They can be used towards wastewater treatment, unlike other methods of alternative energy. They're less dependent on renewable energy technology, and they can also rebuild fuels, and this is so important in helping maintain our planet in human livable condition. You don't want to see this gone. Some limitations of this potential research is that MFCs that were tested all used rumen, thus Geobacter sulfurdosin's bacteria, but not other types of bacteria like Shionella monadensis that are also commonly used in MFCs. It was also not tested whether more saponin addition correlates with more productivity, and that must be researched in the future. In conclusion, we should find a direct correlation between saponin addition and methanogenesis inhibition. In the future, it will be important to research what were the limitations of this potential research. This means testing the effects of saponin addition into MFCs that use other types of bacteria like Shionella monadensis. Testing whether more saponin addition correlates with more productivity, and if productivity did not reach our goal levels, then how do we make it reach them? Lastly, the abstract. Let's use this as an opportunity to recap on the entire presentation. Microbial fuel cells are being researched for their potential of being used as a partial solution towards combating over-reliance on fossil fuel-based electricity in an effort to alleviate the effects of climate change. These MFCs are plagued with problems such as low power generation and the inability to scale up to industrially relevant capacities, and additionally, methanogenesis, the process in which methane production consumes electrons. Korean ginseng and Korean bellflower roots have shown to inhibit methane production and consequently increase in electricity generation in MFCs. The active components in both ginseng and bellflower roots are thought to be saponin. However, saponin has not shown direct methanogenesis inhibition, but has been associated with indirect methane decrease in open systems, rumen of cows, such as less methane production per animal body weight. The MFC anode chamber is a closed system for anaerobic fermentation, and methanogenesis occurs near. Thus, methane inhibition is expected with saponin supplementation based on the fact that it worked with Toraji. However, this has never been directly proven, and this research intends to directly prove that the inclusion of saponin in the anode chamber of the MFC will decrease methane production and improve electron availability for electric power generation in MFCs. That's the end. These are the references.